Welcome, everyone. We're just going to give everyone a minute to get logged in and started, and then we'll begin the webinar. Okay, I think we'll get started then. Welcome, everyone. Happy St. Patrick's Day, if that's something you celebrate. And I'm glad everyone's joined us today. My name is Susan Law. I'm the Knowledge Mobilization Lead for Long COVID Web, and I'm also an Associate Professor at University of Toronto. And we have a terrific couple of speakers today. We're having a bit of an issue having one join us, but we'll try and resolve that as we get started here. I'm going to start with a few preliminary slides. First, the land acknowledgement. We acknowledge that oops, sorry, just jumped screens there. We acknowledge that we live, work, learn, and gather on the ancestral, traditional, and unceded lands of the First Nations, Inuit, and Métis peoples across what is now called Canada. We honor the original caretakers of these lands and waters, and we recognize their enduring presence, languages, laws, cultures, and sacred relationships with the land. We also recognize that acknowledgement must be accompanied by respect, responsibility, and ongoing action toward truth and reconciliation. The purpose of the Long COVID webinars, just as a reminder, is first to share evidence-based information, research results relevant for decision makers, researchers, clinicians, patients, and families, and the general public. Secondly, to provide ongoing updates, spotlights on emerging evidence, and scientific information about the activities of our national network. Thirdly, to contribute to reducing and managing misinformation about long COVID. We do have a code of conduct. The link to that is at the bottom of this slide. And essentially, we value highly engagement around its science and its interpretation as well as application. A commitment to reasoned discourse, as stated in our code of conduct, comes with the responsibility for all of us to ensure that any comments or questions remain within the spirit of positive, constructive, and respectful discussion. So we welcome any and all questions and comments in this spirit. Thank you. Your cameras and mics are off, but you can post your questions in the chat. We will pause for Q&A after each speaker. And actually, if you could put your questions, please, in the Q&A. The chat, you can chat with each other in that function, but the Q&A we will reserve for questions for the speakers. And we'll do our best to review and address as many of those as we can. These sessions are recorded, and we do have permission from our speakers to post these online following the webinar. And we do welcome your thoughts about how these sessions could be more useful and helpful to you. So please take 2 minutes to complete our very short feedback survey, which we'll post. And this is the QR code for that survey. So thank you. I'm going to stop sharing now and welcome Dr. Peter Novak, who's joining us as an Associate Professor of Neurology at the Massachusetts General Brigham and Women's Hospital in Boston, Massachusetts. So thank you very much for joining us, Dr. Novak, and I'll turn it over to you.
Thank you. Can you see my mouse moving?
We can't see your slides yet.
Yeah, thank you. So I'm Peter Novak. I'm a neurologist at Mass General Brigham, and also my research interest covers the long COVID chronic fatigue syndromes. And today I will talk about update about the RECOVER initiative and long COVID. So this project called a platform protocol for evaluation of interventions for autonomic dysfunction in long COVID was mandated by Congress of the United States.
Excuse me, Dr. Novak, we don't see your slides, so I don't know if you've shared them.
Oh, yeah, I do.
Thank you. There we go.
Oh,
thank you.
Sorry. Yeah.
That's
So
perfect.
again, this initiative was mandated by the Congress as a response to COVID pandemic, and it is very complex trial which is funded by the NIH, and it is managed by Duke University. There are a number of the people involved in the structure. So Tae Chang is with me. We are PIs. Tae will talk about a few minutes about Efgartigimod study. And Pam Taup also, she is PI of the Colanor subset of the trial. So again, this is really a very complicated trial. It is overseen by NIH, by several agencies. Again, it is funded by the Congress and NIH and managed by Duke University. And also what is very kind of typical for this, that it is overseas not only for the scientists, researchers, but also by the patients, which also took part in the design of the trial. So the study is testing three hypotheses. It is number one is that IVIG is effective in improving symptoms at 9 months. Second theory will be tested is that ivabradine will improve symptoms at 3 months, and then coordinated versus usual non-pharmacological care will be also effective. So the IVIG is based on the theory that long COVID is due to autoimmunity or abnormal inflammatory response or sequel of the COVID infection, which will affect the immune responses afterwards. And this is based on several open label or anecdotal studies that show that IVIG can be effective in long COVID. Ivabradine portion is based on the fact that patients with long COVID and POTS, they have orthostatic tachycardia, and ivabradine was shown at least in one good study. Actually, it was a study designed by Pam Taup. She is a PI, that ivabradine improves symptoms, both subjective and objective in long COVID patients and POTS patients. So coordinated non-pharmacological therapy is essentially supervised non-pharmacological therapy. Patients are instructed to perform several activities, and they will be managed to make sure that they will be very active. They have several activities will be controlled, for example, by the phone calls. They will get some devices to, for example, to monitor number of steps, Fitbit device, etc. So this has 4 arms, the study. So IVIG, ivabradine, and each has placebo versus a real medication. And again, it is furthermore divided into coordinated versus usual care. So again, those are pharmacological part, and this is a non-pharmacological part. So by that way, it is a multifactorial design. So it is cost effective because more than one intervention can be evaluated. And so this is a study design of IVIG arm. So patients are screened, and if they are satisfy the criteria for the study, they are randomized to 1:1:1:1:1. Again, this chances to be randomized to IVIG versus placebo is 50%. And also they will be assigned randomly to usual care or coordinated care. And the treatment duration is 9 months. And then it will be followed by evaluation at the end of the study and another one after a few weeks later on. Ivabradine arm has a very similar study design, but it is only shorter. So typical duration is 3 months versus 9 months of IVIG. Otherwise, it is very similar. So non-pharmacological intervention is the same. It is usual versus coordinated care. And for coordinated care, so again, it is a type of form of more supervised physical therapy or active physical therapy. Now, patients are really encouraged to have certain level of the fluid, salt intake. They are supposed to use abdominal compression for orthostatic improvement. They are encouraged to do physical activity, exercise, using yoga mat, stretch bands, but again, it will be all supervised. They will be checked frequently, blood pressure, heart rate responses. They will get blood pressure machines. They're supposed to do like a weekly logs regarding her physical activity, making sure that they will be active. And they will be again they are with relatively short or frequent connection with coordinators, which will monitor the activities. So this is more detailed study activities for IVIG. So it will be screening, baseline visit, and visit evaluation at 3, 6, 9 months, and at the end. And for ivabradine, it is similar, but only shorter. Evaluation will be done at months 1 and 3. Inclusion criteria are typical for the POTS patients complicated with long COVID. So again, those are adults, and they are supposed to have previous confirmed COVID infection. Although they were allowed to have few of them also have the non-confirmed, mean without any PCR or antigen confirmation, but only minority of this patient will have non-confirmation. So again, most of the patients were supposed to have either PCR or antigen positive confirmation. They are supposed to have moderate autonomic symptoms defined as a COMPASS score. COMPASS is standard questionnaire for evaluation of the autonomic symptoms. And they are supposed to have orthostatic tachycardia, which is part of the postural tachycardia criterion. So it is an increase of the heart rate 30 beats per minute or more within 10 minutes of standing or during the tilt test. And for ivabradine, the criteria are a little bit less stringent than for IVIG. The assumption is that patient with on IVIG will be more sicker. That's why they will probably require more autoimmune intervention. That's why the reasoning was or criteria were stronger for IVIG. So again, a patient with ivabradine, they will have less severe POTS than patient with IVIG. And it is a phase 2 study. About 200 patients will be in each arm. So it is IVIG or ivabradine. Up to 75 sites in the United States was initially designed. Primary outcome measure is the orthostatic hypotension questionnaire difference from the baseline. So essentially, patient asking how they react to orthostatic stress, how they feel. So this is a primary outcome. It is based on another study that showed that this question about this questionnaire is quite high level of the validity, and it is good for the clinical trials. And there will be a number of secondary outcome measures, including the COMPASS. It is Composite Autonomic Symptom Score. So it is standard survey for evaluation of autonomic symptoms. And there were some other scales like Vanderbilt Orthostatic Symptom Score Scale, ambulatory blood pressure and heart rate measurements, and also 6-minute walk test. And everything will be different from the baseline. And there will be also questionnaire for the evaluation of the symptom burden and general quality of life, and also the quantitative evaluation, including wearable devices, measure the heart rate, step count, daily activities, essential quantification of daily activities. Subset of the sites, they will perform autonomic function testing, including the head-up tilt test, Valsalva, the breathing, which are standard testing for autonomic dysfunction. And if they will, some of them also will do skin biopsies and transcranial Doppler as a part of the tilt test. And the reason why only subset sites do autonomic testing is because not everybody has autonomic lab available. And there will be collection of the specimen, blood, and stool for later analysis. The decision was made not to analyze particular biomarkers at this time because of heterogeneity of the long COVID, and there is no reliable biomarker yet for long COVID. That's why the specimen will be mainly stored for further analysis. And the dose of IVIG will be 2 grams per kilo monthly. It will be typically divided to weekly dosing. Patients also getting saline as a premedication because to decrease the risk of the side effects. And regarding ivabradine, typical dose is 5 milligrams twice a day, but this can be adjusted based on the patient resting heart rate or patient responses. For IVIG, investigators, they have a lot of flexibility in terms of the scheduling of the dosing, as long as they will keep total dose is 2 grams per kilo monthly. And also for intensive non-pharmacological care, patient may get abdominal binders, compression stockings. They're supposed to do supervised exercise rehabilitation. They will get nutrition counseling and other supervised activities to assuring that they will really be compliant with the recommendations. And the study started 2023, and the study is doing extremely well. So this is the recent update. So ivabradine study, a portion already was closed to analysis. The database has been locked. So now it's in the process of statistical analysis and final evaluation. So the ivabradine was essentially closed. IVIG arm is still ongoing. We expect the last patient will be completed within a few weeks, and we expect to have results probably in end of the October or early November. So for ivabradine, there were 48 sites. And enrolled number of the 181 patients. First patient 22, last patient last visit was December last year. Database was log 26. So again, we are anticipating to get results in days, considering ivabradine arm. And I will stop over here. And thank you.
That's terrific. Thank you, Dr. Novak. Yeah, we have a couple of questions, so I'll encourage anyone else to please post any questions for Dr. Novak into the Q&A, the boxes there. So the first question is, if someone has been on ivabradine for 3 years, but their heart rate data appears to show that it's become less effective, and this particular person is using a Garmin heart rate monitor, do you know if this is common, and is there anything that can be done about this?
Well, it is not unusual to see decreased responsiveness to medication, not only ivabradine, but in general for any medication affecting cardiovascular system. So the first step is usually to adjust medication if possible, to change the dose, and again, it's not helping, and still patient needs intervention, maybe switching to other medication, like simply like with similar effect, like beta blockers or calcium channel blockers. Yeah, it depends on the context. Sometimes helps if this is still talking about POTS patients. Sometimes helps to add another medication. There are so many possibilities. So again, it depends why patient not responding. It is because of progression of the disease or cardiovascular system change responsiveness or something else going on.
Okay, thank you. The second question is, someone is curious if all study patients do not have hypertension regarding the salt loading.
But they might actually. There's one form of the POTS is called hyperadrenergic form of the POTS, and those patients, they might have hypertension. And of course, for this subtype of the POTS, the salt loading should be less intense, if at all. So again, for most patients have this hypovolemic or form of the POTS associated with low blood pressure. For those patients, salt loading, high salt intake is usually very beneficial. But again, for this hyperadrenergic variant, actually should not be done or with moderation.
Okay, thank you. Another question, have you been screening for post-exertional malaise, PEM, within the PASC patients, especially those who are in a coordinated care arm?
Not particular for this particular clinical trial. There are no specific questions for post-exertional malaise, but we know that clinically this is a very common complication of the long COVID or dysautonomy, also the POTS.
Okay, thank you. And someone says, as you have mentioned, getting a manuscript together in January, can you share any early readouts?
I wish I could. Well, again, we are expecting results of ivabradine in, I would say, probably a couple of weeks.
Okay, thank you. So stay tuned. And Angela, you have a question or comment?
Thank you. Thank you, Susan, and thank you, Peter, for sharing this. In Canada, we actually have clinical guidelines on the treatment of POTS. And so many of our patients are sort of treated according to those guidelines. And ivabradine is one of the treatments. So it's beta blocker depending on, their blood pressure and pulse. Is RECOVER planning to test other therapies that are in those guidelines?
Not as far as I know, not particular for the POTS. The study only checks ivabradine versus IVIG versus non-pharmacological approach. So not additional intervention is planned for particular for autonomic manifestations of the long COVID.
Thank you. There's another couple of questions here. If IVIG is effective, how long until it's available to patients? I guess that would be in your jurisdiction, or is it already?
Can you please repeat the question? So how long it will be available, or how to say?
Yes, yeah. When will it be available to patients, I think?
It is available even now in the United States. We are using it for years for post-infectious syndromes, for long COVID, chronic fatigue with dysautonomia. We are using it off-label, so it is sometimes difficult to get insurance approval. But for off-label, again, we are using it for a long time.
Mm-hmm. Okay. And someone has asked if there are any improvements in the IVIG arm yet, but I'm assuming that your results will have to wait for your results in a couple of weeks.
So we cannot tell because it is double-blinded placebo-controlled studies, but we know from anecdotal or open-label studies that in some patients IVIG can be very effective. But again, those were limited open-label studies. We did one study actually published like few weeks ago. It was open label, but it was controlled study that IVIG is effective in autonomic inflammatory neuropathies, but it takes a long time to get a consistent improvement. And improvement is, I would say, moderate. It is not, I would say, miracle treatment, but it appears to be effective. I've been using for quite a long time.
Okay, thank you. I see a comment in the chat. We are asking people to use the Q&A box that's at the bottom of your screen, but I will grab this one from the chat. Why do you prefer transcranial Doppler instead of extracranial method, which was developed in the Netherlands?
The reason is because it is much easier to fix the probe on the skull. Over here, you see this 3D this headband, which is once you will fix it, you can use it for a long time, can monitor the blood flow in the MCA, and we use it as a part of autonomy testing, doing the TIL tests. So it is mainly is the technical reason. I know that Susan is using it in the Netherlands. She is using the carotid artery, but it is more difficult to use it. I mean, you have to have a very good skill because you don't have this support from the skull. So it is mainly technical reasoning behind it.
Okay, that's very helpful. Thank you. That's the end of the questions that we have. If anyone has any other questions, please post them now. Is there anything else you wanted to comment on, Dr. Novak? Because we have a few more minutes for you.
So we are very enthusiastic, and we really are hoping that IVIG will be successful because it will be very good proof that long COVID has autoimmune or inflammatory component. Again, long COVID is a very perplexing disease, so with so many manifestations. And there are still different theories. So again, this is a very important trial because it is not only the biggest, it is the first ever trial looking at autonomic complications, any post-infectious disorder. So it will be very important, it will be positive. It might change the way you are practicing medicine, not only for long COVID, also other post-infectious condition, maybe also in chronic fatigue. So we really hope that trial will be successful.
Okay, terrific. Thank you. Well, I'll just say thanks again for joining us today, and I wish you all the best in the remainder of your study with the analysis and the reporting. So thank you for sharing all of that today.
Thank you.
Now, I am delighted to welcome Dr. Tae Chung, but I see that he's just left the room. So I'll just check in with my colleague Karen if he's still...
Yeah, he's having a crash on his Zoom, so he's trying to join from his phone.
Okay, so we'll just wait a second. We can answer perhaps another question that's here for you, Dr. Novak. Is it too late to use IVIG treatment if one has long COVID for many years?
Actually, we think that no, because this is one of the issues we look on a few years ago, and our colleagues in rheumatology, actually, they did show that even, for example, we have a well-defined autoimmune condition, for example, lupus or rheumatoid arthritis, for many years. So still, immune intervention can be effective even after many years of the disease. So we think it could be something similar. So even for years after the post-infectious disease, you still should be able to respond with immunomodulation with IVIG or even other conditions. So durations does not seem to play a significant role.
Okay, excellent. Thank you. Thank you so much. So we'll end it there. Oh, we have one more, and we still have a minute or so. So is it possible that some, most, or all of the long COVID symptoms are an ongoing process that could be correctable rather than permanent damage?
Well, of course, it is possible. Many patients without any the evidence of the structural damage, for example, without any pulmonary or cardiovascular abnormalities. So those patients, we think they do have autoimmunity abnormalities. So simply immune system is remembering previous conditions. So the whole the idea using IVIG is not to only suppress immunity, but to restore, because IVIG is also immunomodulatory. It modulates B T cells. Actually, this is the main theory about using IVIG, is to modulate or to reset immunity so it will recover completely to the previous state.
Okay, thank you. Very helpful explanation. Thanks so much. So we'll turn now to Dr. Tae Chung, who, despite numerous technical difficulties has joined us, so thank you for hanging in there and welcome to this session. Dr. Chung is director of the Johns Hopkins POTS Clinic program. He's associate professor of physical medicine and rehab at the Johns Hopkins University in Baltimore, Maryland. So welcome, Dr. Chung. I'll ask if you can now share your slides and turn it over to you. Thank you.
Thank you. Thank you so much. I've been having a lot of technical troubles and appreciate for your help and support. How much time do I have for this?
We have 30 minutes left.
30 minutes? Okay.
Are you able to share your slides from your phone or?
I actually I don't think I can. Can you share this for me or?
Yes, if they're not too large a file, you can send them to us and we can
I just
share
send
them.
it to you.
Okay,
and it shouldn't be that,
Yeah. Is it okay to send them to you, Karen or Nick?
Yes.
Yes.
Please send.
Great. So we'll just wait for those to get loaded.
Yeah. Again, apologize for technical trouble. Like it took a long time to join the meeting, and then my laptop computer is sold. If I stay in Zoom too long, then it crashes. So that's another problem.
Oh dear. Okay, you need to put a new computer in that next grant.
Right. And I think I told you, but I'm actually leaving Johns Hopkins. Tomorrow is gonna be my last day at Johns Hopkins. So I didn't bother to turn in my laptop computer.
That's okay. Perhaps you can tell us just where you're going, so
Sure.
people know, and then while
Yeah.
the slides are
I'm
getting
actually,
loaded.
thank you. Yeah, I'm actually joining. It's called Shirley Ryan Ability Lab. It's the independent rehab hospital, but it's going to be part of Northwestern University in Chicago. And I'll be opening the same thing, POTS Dysautonomia Clinic, in a little bigger place. So I'm excited about joining them. So hopefully I can continue to collaborate with you.
Congratulations.
Yeah. Did you get my slides or?
Not yet. Still waiting.
Okay.
I received the slides, so I'm happy to share.
Oh, thanks, Fish.
Oh,
That would be
perfect.
great.
Thank you so much. I really appreciate it.
Oh, no worries. So please let me know if you can see them. And
Okay.
That's
Perfect.
great.
Thank you.
Thank you.
All right. Well, thank you so much. Again, my name is Tae Chung. I'm currently at Johns Hopkins, moving to Northwestern as of tomorrow. So I'll be talking about the study that we completed actually last year. And this is a clinical trial for long COVID POTS or post-COVID-onset POTS. And the intervention is a new medication called Efgartigimod. The next slide. So, so this was a randomized, double-blinded, placebo-controlled, total of 36 weeks, and phase 2 study. And I believe this is actually the first FDA-regulated trial on POTS indication. Prior to COVID, there had been some trials, like mainly investigator-initiated trials, but not really multicenter, double-blinded, randomized clinical trial with the goal of getting the FDA approval. This was the first time. Was started in September 2022, and I still really appreciate Argenx Pharmaceuticals who sponsored this trial. POTS, as you know, is not a really easy target for pharmaceutical companies to do trials on, but that was actually early in COVID pandemic, and long COVID has been emerging as a really global health threat, and the leadership team in our pharmaceutical, Argenx Pharmaceutical, really teamed up to really start this FDA trial. Around the same time, shortly afterwards, they also started open label extension of the same trial. They actually have a little different trial, registered as a little different clinical trial. Now, both completed around the same time in 2024. The original study completed in April 2024. Because the original study didn't show clear efficacy, they terminated open label extension in the middle of the open label study. So next slide, please. So basically, go back to the previous one, please. The rationale is that as many people agree, POTS we do think is autonomic nerve dysfunction. There are currently a few different theories about why POTS happens, and one of the main thing is vasomotor denervation, which results in excessive blood pooling below the heart and lung chest segment that causes severe cardiac preload, and that's causing severe fatigue, brain fog, and exercise intolerance. At the same time, that's causing all these hyperadrenergic symptoms through baroreflex and causing tachycardia, insomnia, and GI troubles. So particularly in post-COVID-onset POTS, we thought that this is more likely, inflammation to postganglionic sympathetic vasomotor nerve. Early in the pandemic, there have been some evidence showing pseudomotor denervation, sympathetic nerve denervation in POTS or long COVID patients. And also, because it's a post-COVID and post-infectious onset, we thought it was more immune-mediated, autoimmune onset POTS. At this point, I'm sure Dr. Novak briefly explained, there's no really reliable biomarker for autoimmunity in POTS. POTS is a heterogeneous condition. Some people may get POTS from some other genetic or metabolic reasons, but the subset of POTS that are autoimmune, we don't have any biomarker to identify them. But somebody who are healthy prior to COVID infection and dramatically develops POTS after COVID infection, we thought there's a more likely, there's a pretty good chance that these are autoimmune autonomic inflammation that's causing POTS. So we targeted these people using the new immune modulating medication called Efgartigimod. And also people like Dr. Peter Novak and other people have already reported IVIG on a post-COVID POTS patient with some notable benefits. Next slide. So Efgartigimod is one of the new class of immune modulating medication called FcRn receptor antagonist. Basically, this receptor is a receptor that recycles immunoglobulin at the endothelium. Now, by blocking this receptor, the endothelium is not recycling the immunoglobulin, especially IgG, and it goes into lysosomal pathway. So basically, by blocking FcRn receptor, people have shown that the serum IgG level goes down, but it only specifically works for IgG, and human IgA and IgM level remains intact. So it's a mild immunosuppressor. A lot of people, because of intact IgA and IgA or IgM levels, they actually respond to vaccination and some protection against some other infectious agents. Now, because we think that most autoantibodies are IgG mediated, we wanted to target. We need to try this Efgartigimod for post-COVID onset POTS patients. Although we didn't have any clear autoantibody biomarker that identifies autoimmunity in POTS patients. Next slide, please. So the study design overall, again, randomized double-blinded placebo-controlled parallel group, total 30 weeks, phase 2, and it's conducted in 11 sites only in the United States. And they're randomized to 2-to-1 design, meaning participants are twice more likely to get the drug rather than placebo. And placebo is basically saline-based. It's not a huge amount of hydration. It's only just about 200 cc's. So we didn't expect any blood volume expansion benefit from the placebo. And Efgartigimod's dose at 10 milligram per kilogram is basically based on their body weight. And it was actually every week for 24 weeks, meaning 6 months. So the participant had to come into the site every week for 6 months, which is very involved study if you think about it. And the total 30 weeks includes actually 4 weeks of pre-screening and 24 weeks of active intervention and 8 weeks of follow-up period. Next slide. So just going to the major key eligibility criteria, inclusion obviously is a history of COVID-19 infection. Now, initially the study, we required the confirmation of COVID-19 infection by PCR. But as you can remember, as the pandemic kind of continues, a lot of people didn't really get PCR testing, especially after the antigen test became available. So later on, we had a major protocol change that included some people who got COVID, who has approved of COVID infection by home antigen test or some clear documentation from doctor's office and so on. Also, I didn't include details, but of course we had to show that the patient had POTS by tube-table test or active stent test. And as you may know, the clear tube-table test criteria being more than 30 BPM increase from supine position without blood pressure changes. And also we need as a part of eligibility, they need to prove that they had ongoing POTS symptoms that lasted at least 3 months after COVID infection. Now, of course, that turns out to be slightly difficult because we started the study in just middle of 2022. So that was actually really the beginning of long COVID pandemic. And a lot of people didn't have that more than 3 months of kind of symptom persistence. Also, we wanted to make sure, because we didn't have a clear biomarker or outcome measures, we wanted to make sure that we recruit participants who have very severe autonomic symptoms. And we determined the severity of autonomic symptoms based on COMPASS 31 score. For those who are not familiar with this questionnaire, this is pretty much almost only autonomic symptom questionnaire that are validated and used for a lot of other autonomic diseases in the past. COMPASS 31, by the way, is not specifically designed for POTS, but at that time, that was the only question that are available that's related to any autonomic nerve symptoms. Now, exclusion criteria, the main thing was to exclude any autoimmune diseases or overlapping other autoimmune conditions, especially anybody who were exposed to immune drugs were all excluded. We also wanted to make sure that this is actually also fresh new onset POTS after COVID infection. So anybody who had COVID, who were diagnosed with POTS prior to COVID infection were all excluded. And not only POTS, just because there's a lot of overlap between other conditions such as ME/CFS, EDS, or MCAS, we wanted to exclude all these people who were diagnosed with a similar overlapping condition with the POTS, so that there's no other kind of signal coming into this trial. Next slide. So again, this over 24 weeks, it's 2-to-1 design. Efgartigimod, twice more likely than placebo for 24 months and 8-week follow-up. Next slide. I know it's just a little kind of a small wording, but basically total, let me say, total 83 participants were kind of screened for eligibility, and over 53 participants were randomized. And if you go all the way down to intervention, the left side of the graph is people who got the Efgartigimod, the drug, and the right side of the panel is a placebo. And I know it's a little kind of small, maybe it's too small to look at, but the completion rate is actually 83% for the Efgartigimod group, and then 88% for the placebo group at the end of the intervention. So the completion rate is actually pretty equivalent. And again, I want to also remind you that this was actually a very, very involved study. This is actually every week for 24 weeks, 6 months IV infusion. So the patient had to get IV infusion. So given that, we felt that 80% is actually really nice compliance, and very few people have dropped off the trial for various reasons. I mean, not only potential AEs, but also, some other kind of logistical reasons and patient go on vacation, relocation, and other things too. Next slide. So, like I briefly mentioned, unfortunately, the main outcome measure, which is COMPASS 31, the main outcome measures are actually two questionnaires, which is COMPASS 31, and MAPS stands for Malmo POTS. A lot of people call this Malmo POTS because it was developed by Artur Fedorowski in Sweden, in the Malmo, Sweden, and this was the kind of probably the first questionnaire that are validated for POTS. So we kind of use these two questionnaires of main outcome measures. And obviously, main outcome measures a reduction of baseline to week 24 in those kind of questionnaires. And I can show you the kind of graph in the next slide, but both didn't show, statistical significance between these two, scores. And in fact, interesting thing is that both groups showed actually very significant improvements in both kind of symptom scores. Next slide. So this is actually graphical kind of representation of this main outcome. So the upper panel is a COMPASS 31 score. The lower panel is a Malmo POTS, and the green line is Efgartigimod, and black line is the placebo group. You look at the COMPASS 31. The starting point, the baseline, again, like the little small letter, but the left-hand side is a baseline, and all the way down to the week 24. And you can see actually the reduction of COMPASS 31 scores over time in both placebo and Efgartigimod group. The same thing, similar pattern you can see in Malmo POTS score as well. From baseline 0, you see a reduction in the scores in both groups by week 24. I guess a little difference you can see Malmo POTS again is COMPASS 31 has never been validated for POTS. Malmo POTS, whereas the questionnaire is more specific for POTS score. And in Malmo POTS, you see a little kind of difference, little separation in the middle, although these patterns are not statistically significant. But you see a little different pattern. But overall, also another thing that I wanted to mention is that this reduction in the score, like almost 10 points in COMPASS 31, almost 20 points in Malmo POTS, are clinically meaningful reduction. This is not something that you see, day-to-day kind of, variations. So these are actually clinically meaningful reduction in those score, which we thought is kind of really interesting. And also, again, I would like to remind you that this is a 2-to-1 design. In the placebo group, total we only randomized only 17 participants in this trial. Next slide. Now, interestingly, we had various different secondary outcomes, and a lot of secondary outcomes showed very similar patterns. They both kind of got better, but nothing really significant. Significant in the statistical measures. I'm sorry. So those secondary outcomes includes change in the heart rate on the teal table. Obviously, both got better from the baseline, but between the groups, no difference. And other secondary outcome measures, including PGIC, Promise Fatigue Scale, and 6-minute walk test, didn't show a lot of difference. Now, this is a more secondary analysis. We actually measure some inflammatory biomarkers such as cytokines, like interferon levels, and they didn't show any difference between the baseline and week 24 or between the two groups. They've seen actually some mild reduction in von Willebrand factor and BCAM, which is some clotting factor, which I think is kind of interesting, but we still don't know the meaning of it. Next slide. Now, obviously, okay, Yeah, obviously the total IgG level has significantly reduced in the Efgartigimod group, whereas the placebo group didn't show any difference, which just shows that this drug did what it's supposed to do, reducing the IgG level. Next slide. Now, more importantly, the safety profiles are actually pretty excellent in both groups. The adverse events were very comparable or similar. Actually, the proportionally fewer participants in the drug group actually reported drug-related AEs than the placebo group, and no major SAEs or fatal drug-related AEs are reported during the study. Common adverse events include just fatigue, headache, and nausea, but drugs are really well tolerated. We did see some UTIs, it is immunosuppressant, but nothing really significant between the two treatment groups. And these safety profiles are very similar to those reported in other trials targeting myasthenia gravis or CIDP. Those are safety profiles in the previous trials in other disease conditions are very, very similar to what we saw in this post-COVID trial. Next slide. So conclusion, both efgartigimod and placebo improved over 24 weeks in this trial, and not only primary outcomes, but also all the secondary outcomes, including heart rate changes and 6-minute walk test and other kind of functional outcomes as well. And also no signals are observed. Next slide. So just briefly, I think one of the really important take-home messages is that there's a huge placebo effects in this population. I think this is something that we saw similar thing in IVIG trial. It's called iSTAND trial before COVID pandemic. There was one small IVIG trial. We actually saw similar huge placebo effects. So I think this is a really important take-home message from this trial going forward, designing future trials that's targeting POTS. And also, I think this really study shows there's a desperate need for better biomarkers, especially that identify subpopulation of autoimmune onset POTS. Again, you probably noticed that in the eligibility criteria, we didn't include any inflammatory markers or some related autoantibody markers, because we thought that by including those autoantibody markers, I think we can probably select some other disease condition. But we do think that there's probably novel autoantibody marker that we yet discovered. And another really important thing about this clinical trial, although it didn't show the efficacy, we already collected all the blood markers, and this is all potential to discover really good biomarkers. Obviously, there's a really good, better need for better outcome measures specific for POTS. We actually learned that Malmo POTS questionnaires are pretty decent. And going forward in the future trials, I think Malmo POTS is a really nice, scores that are pretty reliable. We also, another thing that I've been thinking about is that we did 6 months trial because it takes a lot of time for these small autonomic fibers to recover. And actually, we have quite a few patients who reported much better improvement in their symptoms during the open label extension, meaning like 7-8 months into it. But unfortunately, when they closed the phase 2 trial, they terminated open label in the middle of the trial. Open label extension was supposed to be, I think, another 6 months to a year. So I wonder if they completed open label extension, I wonder if the results could have been different. But of course, we are open to any other suggestion. I mean, again, we learned so much out of this clinical trial. I'm still appreciate Argenx, taking the initiative of this really important clinical trial. And this whole results will be, we actually submitted for publication, so still waiting for the review, peer review at this point. All right, I don't have a lot of kind of time limitation left, but more than happy to get any comments or questions. Thank you so much.
That's terrific, Dr. Chung. Thanks so much for your presentation. We do have some questions, and the first one is, how are you going to investigate the reason why both groups showed significant improvement? What is it that you suspect helped the placebo group?
What is what? What is the second one? Question?
What is it that you suspect helped
Oh,
the placebo group?
yeah. I mean, so there are so many reasons, to be honest. To answer the first question, I don't know how to investigate why it is. But the first, well, my suspicion actually has been that there are actually, people that I recognize, um, that some long COVID POTS patients, there are some patients who actually got better spontaneously. These are the people who actually showed monophasic course. They got really severe POTS, and over usually several months, they just got better. Whether that's kind of Guillain-Barré type of autoimmune but monophasic phenomenon, or it's still possible that COVID virus can directly impact autonomic nerve and then show just kind of infection related kind of nerve damage and showing the monophasic course. And that's actually why if you remember the graph in the mammal POTS, there's a little split in the Efgartigimod and placebo group. And I wonder if that's potentially why, where in the monophasic group has a little more linear recovery, whereas Efgartigimod group actually shows a little, kind of S-curve kind of a, recovery pattern. And maybe it's possible if you had more participants, we could show some meaningful difference there. But at the end of the day, I think I do notice that there's still very big placebo effects, and there's a really need for good biomarker that can separate these people.
Thank you. I see that Dr. Angela Chung has her hand up. Go ahead, Angela. Oh, we can't hear you.
Thank you, Tay, for presenting. So I'm just wondering, do you think there are co-interventions in the trial? Like, do you teach patients to rest and pace no matter which group they are in? And do you think that those kinds of co-interventions may have affected your results?
That's a great point and great question. I wish you asked that question before we designed the study, but of course, we didn't. And also another thing, actually, going back to the point is that POTS patients, as you all know, they have a very variation in their symptom severity almost daily basis, and it's kind of hard to factor into that variations and maybe co-intervention pacing so that they can somewhat controlled in their symptom variation. I think that'll be really helpful and something to think about for the next trial. And another thing just to add, like this is probably not directly related to your PM question, but the same company, Argenx, they did on the CIDP patients. And the CIDP, the clinical trial, they didn't have actually also autoimmune biomarker. And they tried what is called placebo run-in trial, where everybody got placebo in the beginning, and people who had too much response, they got excluded, and then the less placebo effect participants were included in the actual trial. And I think that's another potential, design we can use on this population, somewhat, less specific in their symptoms and without any good biomarker. I think that's actually not a bad idea. And that's, by the way, the CIDP trial also got the FDA approval with that clinical trial design.
Thank you. This is a question about whether or not you will follow patients longer to see if there are differences long term.
For the clinical trial or just patients in general?
I guess you could answer both.
Yeah.
Yes.
So, again, like that's another really important take-home message. I really think 6 months is really just clinically speaking. I'm sure Dr. Novak will agree with me that it's just really the beginning. If you see any difference, and I just say again, like my theory may or may not be correct, but the theories behind it is that these are small fiber autonomic nerve that are potentially denervated, and then by stopping inflammation, it allows regeneration. But this regeneration will take a really long time because it's a very long axon. Like it's not really surprising to see people getting better, recovering over a couple of years. So I think 6 months was really the kind of shortest time for the trial. So that's actually why I was really hoping that open label extension continued. Then I wonder it could have actually made some difference.
Thank you. Another question, is there any risk for such kind of trial treatments? Is it suitable for long COVID patients with mild symptoms?
Well, as you saw in the safety profile, actually, we really didn't even see a huge amount of kind of infections, although it's an immunosuppressor, because I guess there's probably IgA and IgM there. It's really well-tolerated medication. I don't really see big concerns, safety concerns with the medication per se. But obviously, this is immunosuppressants, so you don't want to do this in healthy patients. Also, at the same time, mild symptoms. Well, I guess it depends on how you define mild symptoms. But one of the main reasons we wanted to screen out mild symptom patients was that we didn't have any good biomarker. We wanted to see somebody who's struggling a lot and getting better a lot. So that was a main reason. But I don't see any major safety concern using it for mild long COVID patient, as long as you're convinced that it's autoimmune.
Okay, thank you. We have a couple more questions. Do you have a couple more minutes to stay?
Oh, yeah, I'm fine. Yeah, please.
Okay, that's great. Good.
Sure.
Did the drop in IgG measured in the study group receiving Efgartigimod use completely reverse back to normal after the medication was stopped?
Yes, they did. I actually forgot how long did it take to kind of reverse it. It would take at least a couple months before it gets to normal level.
Okay, thank you.
Mhm.
Do you have any thoughts on which subsets of long COVID patients might respond best to Vivigard?
Which subset of long COVID? I don't
Do
know
you have
what
any
subset
thoughts
there.
of, which types or which subsets of long COVID patients might respond best to Vivigard?
I don't know if there's any well-known subset of long COVID, official subset, but I'm sure, of course, I mean, somebody who, like autoimmune kind of evidence where like their symptoms kind of flare up and down. It's clearly post-infectious onset. I mean, I don't know if that answers the question really well, but
Okay, thank you.
Yeah.
How long does it take to regrow the fibers?
Well, I mean, we assume that nobody actually really done it on the small fiber, I believe. I mean, Dr. Novak may know about it, but usually this kind of long axonal regrowth happens like, they say 1 millimeter per day or something. And you have to imagine the cell body is right at sympathetic chain ganglia right next to kind of spine. And then it can innervate to all the way to the distal limbs. So just imagine how long it would take to do that.
I would just
Thank you,
help you
Doctor.
that it will take about approximately 1 year to grow the axons from the back to the distal leg if there's ongoing damage. I think in diabetics or some disease was described up to 2 years. So it is between 1 to 2 years.
Right. And also
theoretical time of the regrow is based on the axonal transport.
And also, in the peripheral neuropathy world, as you know, the patients who are tall and long arms and legs, it takes a much longer time. Actually, they do have a more poor prognosis when they have axonal injuries. So we do think that it's probably likely same. Or if anything, it can be even like slower because the bigger myelinated fibers, they get all the nutrients to guide their axonal regeneration, but small fibers don't get enough nutritional support from the Wallerian cells, and I guess a lot of cells probably don't even get regenerated, they just drop off, or could be even slow. But I couldn't really find any really reliable kind of basic science data how fast the small fiber regrows. Do you know anything by any chance, Peter?
Well, again, in theory, between 1 to 2 years, but we did one study just published a few weeks ago, but we look on the post-infectious disorders, and average duration of the IVIG improved consistently symptoms was 2.2 years. So it takes a long time.
Right.
So again, it took 2.2 years to see kind of consistent response or improvement in autonomic symptoms.
So,
Okay, thank you. Two more questions then. Do you plan any trials on immunodesorption?
immunoabsorption. I mean, personally not, but I don't know, Peter, you probably remember that I know there's a like a Scheibenbogen. I think in Germany there was a kind of similar trial, but I believe I'm not familiar with the immunoabsorption trial, but my understanding is that there is a trial if they know the pathogenic autoantibody, but we don't know that.
Yeah, I'm not sure, but I know that, for
Yeah.
example, for long COVID, we check trials few years ago. It was almost 400 trials. So, there's a lot of activities going on. I'm not sure how many were published, but there are still ongoing so many trials targeting different aspects of the disease, or immunosuppression, immunomodulation, antiviral, and other things.
Yeah. I mean, again, like the immunoabsorption therapy, my understanding is it only observed the pathogenic autoantibody. And I know in Germany, they did on a cell-train antibody, which is a GPCR antibody. And I know Dr. Chung was there. And as of last time, actually was in the meeting. I don't think the results are available. But,
Okay.
hopefully it might have been published by now.
Okay, thank you. I'll just finish with this question. Why isn't stroke volume measured by ultrasound used as an outcome measure in clinical trials?
Well, I can answer this. It is not that easy to measure stroke volume consistently. Now, we do have algorithm. We measure stroke volume from the continuous blood pressure, but it is not always accurate. And those patients with the pulse, they might be hypovolemic, might be even more difficult to get it. So invasive testing exists. It's very accurate, but it is invasive. It will be not technically possible to use it in everybody. And also, it's always preferred to use subjective reporting over. That's why it's the policy of the NIH to use subjective patients' symptoms the first, although we also use objective markers if possible. But again, as I also and I also try to explain, we don't have good biomarkers of the long COVID and inflammatory neuropathies in general, which makes everything more difficult. So we use proxy, like blood pressure, autonomic dysfunction. But again, still the most important is what the patient will tell you, how they feel, how they respond.
That's true. Yeah, I second that too. Like stroke volume by echocardiogram, I was told by cardiologists is not super reliable. Also, we are not sure if that changes in the stroke volume actually translate into patient subjective symptom improvement or not. We don't know about that too. And I saw one of the comments real quick about related to this question, why not skin biopsy, small fiber density? We actually included in the study design. Unfortunately, purely because of logistical reasons, skin biopsy is really not done for many subjects. So we couldn't even we got just 2 or 3 skin biopsy samples out of the whole 11 sites. So we couldn't really even analyze it.
Okay, I think we'll finish there. We're a bit over time, but thank you everyone for staying on, and thank you very much for your presentations. Very, very interesting. We will post those online very shortly on our website. And I have in the chat there, one of our colleagues has posted these short surveys. So please do take a couple of minutes there to give us some feedback. And I just want to wish you, our speakers, both all the best with the remainder of your work in these studies, and all the best with your transition, Dr. Chung.
Thank you so much. Thank you for inviting me.
Thank you.
Thank you,
everyone.
Peter and Tay, for great talks. And sharing your knowledge.
Thank you.
Take care, everyone. Thanks.